Okay, good evening everyone. Today we are scanning a sample from Giv'on pond, which is another ecological pond about 500 meters from the previous pond that we've scanned last time. But this one is quite shallower, around let's say 20 to 30 centimeters, so I can easily grab the benthos layer, the bottom layer of the pond, and there are many other creatures that dwells there that we haven't seen last time. I'm going to scan two petri dishes today. One for the very big creatures. We are now watching one that's this worm, and then we'll move to the more delicate small creatures. Let me just close the window for 1 sec. Okay, much better now. So let's start with this nice big worm. I'm now looking with the 4x objective, and our field of view from one side to the other, whether you're watching it on a big screen or a smartphone, is around 6 millimeters. So this worm is, if we stretch it sideways, it's around 6 millimeters, even a little bigger, so easily spotted with our eyes. She's calm now, and if we'll zoom on its head, you'll see those two black spots, which are its eyes. We can also see at the base of it like a suction cup that helps it adhere to the ground or other areas. Oh, she starts moving. And if we're examining the center of it, we can clearly see the digestive tract. We are now with dark field illumination, and later I'll move to bright field illumination. We'll see it more clearly. We can also see some ciliates hovering around. And let's zoom out a bit. Here are two hydras. We already seen them last time, but they're also in this sample. For those of you who haven't seen the last chapter, the hydras are a multi-cell creature that's a relative of a jellyfish, and it has venom cells in its tentacles that activates when it senses prey and paralyze it. Then the tentacles move the prey into the mouth, which is in the base of the tentacles. You see they are very contractile. This one is starting to move a bit. They can actually leap on its tentacles, and this one is spread all the way. Here is another creature from the crustaceans. This one's called an ostracod. It's a bivalve crustacean. If we'll wait patiently, it will open its valves. See, and it sends its legs. And this spot in the middle, it's its eye. We'll also see it more clearly in bright field in a second. Let's zoom out. And there was another creature, yeah. This is another crustacean, a water flea. It tends to run away, so we'll do it quickly. You can see its legs inside the body, and this spot, its eye. And like the ostracod, it's a crustacean. Let's move to bright field illumination for a second. We'll have to lower the luminosity quite a bit. Adjust the lighting. Okay, so now with bright field illumination, we can see the details in the water flea. Let's try to add a blue filter so it will be nicer to look at. Yeah, much better. Maybe open this one a bit. Okay. So here is the water flea, and beneath it the worm, flatworm that we've seen earlier. Now you can see the two spots of its eyes, and the digestive tract is clearly visible, just like our guts. And the suction disc. What else we saw? Oh, the ostracod, there it is. This one needs a higher magnification. So I'm moving now to the 10x objective. And yes, we can see its eye now. And that it has two separate valves. It comprises of two separate valves like a clam. It has a nice pattern on its shell. Okay, that sums the big creatures. So now I'll move to a different Petri dish. We'll go back to the 4x objective with dark field. So we can start examining what we've got here, and then we'll move on to higher objective. So now we're just scanning. Let's do it methodically from right to left. Okay, we can already see a ciliate called spirostomum. Here's another ciliate, a stentor. Another ciliate, a frontonid. Another frontonid, very different in shape. Here is one that we've already seen in the last chapter, Loxophyllum. This one I can't judge from here, but it looks like Loxidus. Let's move to a little bit higher magnification. And Yeah, I think it's Loxodus. Spirostomum ciliate, another frontonid, another frontonid. We can see here, this one looks like an amoeba. We'll watch it in much higher magnification later. And this structure in the middle is another amoeba. It's a testate amoeba, which is amoeba with like a skeleton around it, a silicous layer around it. And those are the creatures that live on the bottom layer. Here's another tiny rotifer, Spirostomum. Here is another testate amoeba. Here are some peritrich ciliates. Here is another nice ciliate urocentrum. More testate amoebas. Oh, here is a very nice one. That's a red hypertrich. Quite unusual. Again, we'll see all those in details very soon. Here is another rotifer, a euplotid ciliate running around. More testate amoebas, another red hypertrich. That Loxodus. This one I don't know, we'll see later. A stentor. Another Laxophyllum in its stretch form. And this one seems like a Litonatus. Oh, that's a very large Spirostomum. This one you can even see with your eye. So let's jump to a higher magnification. Sorry for the intense light. Now it's better. Let me adjust the illumination for a sec. And we are good to go. So yes, that's Loxodus. We'll wait till it calm down a bit, and then we can, yeah, now he's doing his normal thing, which is crawling on the surface. Let's see what else we can see. Another testate amoeba, another testate amoeba, another one, and here are those peritrich ciliates we've seen before. I think maybe even let's jump right to 20. Yeah, very nice. Now we can see here is another testate amoeba, an elongated one, and we can see that the structure, this one has a structure that comprises of different aggregates that it collects from the surroundings, like small stones. You see, it's built like a small wall. I'm adjusting the illumination. Here is this red hypertrich that we've seen earlier. Let's see what else we can spot. Okay, that's another amoeba, and this amoeba, as you can see, has no exoskeleton, no protective layer. It's just a blob without any cover. That's another testate amoeba. You see it's much darker and yellower than the others we've seen. That's because it's probably older and some iron or manganese has stuck to its skeleton. Here is a very nice. Oh, it ran away. This one is very fast. It was wow. Got a blackout for a second. But this one is a very nice rotifer. You can see its eyes now, two red spots. It moves up and down within the water column. Here is another ciliate called Frontonia. Oh, it ran away, but we'll see some more later. There are many in this sample. That's a rotifer. We can see its two toes here. And here comes Loxophyllum, one of my favorite ciliates. The ciliates are, for those of you who haven't been here before, the ciliates are a group of single cell creatures that bears cilia, like its name suggests. I love this ciliate, it's very regal. Let's see who else we've got. Here is another amoeba. We'll look more closely later on amoebas with phase contrast illumination, which will accentuate its detail more clearly. And right here we can see a tiny star in the middle. It's an heliozoon. We'll look with it with 40 later. We can see a flagellate here, and this spirostomum just entered our scene. It looks like he has a broken neck, and sometimes they got hurt when I transferred them with the pipette. And here is Mr. two euplotids, very agile. And on the debris we can see a stentorid. A stentor, I remind you, means in Greek a trumpet, and you can see its shape resembles a trumpet. See how it contracted as soon as the euplotid touch it, and if we'll wait patiently it will come back soon. But for the moment we can see how the euplotid exploit their merged cilia to walk on surfaces. Here is the stentor tucked inside, waiting to get out. Oh, and it got back in. Okay, back to the substrate. Here's another spirostomum, much shorter than the one we've seen before. Here is another small hypotrich. testate amoebas. Frontonia, and those lovely red hypertrichs. Let's move to 40 for this one. We'll exploit the fact that it's relatively calm. I'm just now correcting the lens. That looks nice. And you can see it's unusual, uncommon red color. Okay, now it's climbing on the debris, so it's not so clear. Let's see what else we got. I'll go back to the 20. I remind you that if you got any questions, please write them in the chat box and I'll answer them. Here is a nice little euglanid. Oh, this spot is quite empty. Let's see where all the fuss is. Here is another frontonid, quite different than the one we've seen earlier. And Spirostomum, very long and contractile. You've just seen how it contracts when it touches something. Testate amoebas, and this one in the middle looks like a rotifer egg, or could be a tardigrade egg, a water bear egg. I'm not sure. Okay, looks like this spot is very active, the one we've started in. So where was that amoeba? Here, change to 40. I need to adjust the lens color for a second. And the illumination is quite off. I don't know what happened. Oh, because I switched the petri dishes. So now it will be better. Okay, nice. So now we're seeing an amoeba, a naked amoeba, and you can see how it doesn't have a clear shape. It's amorphous without a clear shape, and inside of it we've got lots of crystals. These crystals are some of the characters that help us to identify which species of amoeba is it. And you can see now it sends one of its pseudopodia, false legs, and you can see how it moves very slowly. Yeah, now it's just okay. Now let's try to locate one of the testate amoebas, the one with the skeleton around, the protective layer. This one is an egg. Here is one of the but this one doesn't send any legs outside. Oh, here is the rotifer that we've seen earlier. Okay, let's move to 24 sec, locate one of those testate amoebas, and then we'll switch back. That one, I don't see it sends any legs outside. This one is. Okay, great. So now switch to 40, and for another illumination technique called phase contrast. And I've seen that leg. Where is it? Okay, it's quite shy, so we can't see its leg. But what we can see now are all the bacteria that are on the bottom of the petri dish. I'll move another. Oh, now it's better. And let's see what else we can spot with this illumination technique. Oh, here is that amoeba that we've seen before. We can see now the crystals more clearly. I lower the intensity, and you can see the pseudopodia. This one is quite thick, so it's hard to get a good image of it. But you can see the crystals very clearly. Oh, this one is quite nice. I'll move back to the IC illumination. And I need to adjust the color of the lens because this one is very high in the water. And now we can see an heliozoan. This one is like a star with the radiating tentacles. See all those tentacles, and there are three of them here, and it catches something. It caught something. This one is Urocentrum, another ciliate. So we're just seeing that one of those heliozoans, four actually, 1, 2, 3, 4, and it caught another ciliate, which its name is Urocentrum turbo. So this ciliate is dying now, and if we'll come back later, we can see it digested. Okay, moving to the ground. Need to adjust the color. Better. Okay, let's see if, oh, here are those very, very nice red hypertroph. Just like tiny red animals. A very cool species. And besides of it, we can now see a bottom view of a testate amoeba. It's like a cave. You see that opening? And if I'll go up, you can see all the aggregate particles. And inside of it, we can barely see, but we can, the pseudopodia. Let's see if we move to phase contrast, if it will be more clear. Sorry for the illumination jump. And let's adjust. Okay, we now see the opening of that testate amoeba. But actually the pseudopodia were much clearer with DIC. So I'll move back to DIC. And let's try to increase the contrast more. White shifted. And in the middle we can barely see the pseudopodia and actually the living part of that amoeba. Now it just hangs around and doesn't send its legs outside. Spirostomum. Okay, this one is quite calm, so let's follow him. You can see all the cilia that is around it, and it has a multi-nucleus inside of it. You can see a lot of bulbs inside. Okay, I'm going back to, oh yeah, that's Frontonia, a very nice species. I'm going back now to 20 to see if we missed something. Before that, we just caught that one. It's a scuticociliate. Let's see if we can bump magnification a bit. And this one is scuticociliates. It's a family of ciliates, and we can, oh, it went away, so never mind. Let's go back to 20. This one is a stentor. Here is that nice Laetonatus that I spotted before. Very elegant ciliate. You can see in the middle of it two large nuclei, and we can barely see the micronuclei between those two. Okay, back to 20. We've reached the perimeter here, so let's scan around the perimeter because it's shallower here. Oh, that's unusual for Loxophyllum to float on the upper surface, munching the bacteria there. I'm just scanning to see if we got another interesting species that we haven't covered. So bear with me for a sec. Oh, this stentor is finally out. You can see its distinctive trumpet shape. Here's another opening of testate amoeba. That's a calm one. A rotifer. A rotifer. Oh, that's nice. Now we see Frontonia that's in the middle of division. One of the ways ciliates are reproductive, it's by cell division, and this one, we'll try to track it. Oh, it's gone. Sorry, I've lost it, but as you can see, there was like a belt in the middle of it, which indicates that it's in the middle of division. So one cell divides into two exact copies. Here it is again. You can see it has a distinctive shortening in the middle, and not so long from now it will be two separate cells with exact copy. You plotted, and Loxophyllum. Okay, this one is clearly this testate amoeba spreads its legs, so let's look at it. Why can't I see it now? It's very strange. Maybe it's sending its legs. Very high, so we can't see it on higher magnifications. So back to 20. So it took it back. Sorry, we can't see the legs now. Sometimes you need luck. So I think we've covered everything. Wow, that's a very long spirostomum. Those spirostomum can reach up to 2 millimeters. Let's see how this one is. I'm going to 10x now. So our field of view is 2.2 millimeters, and this one is, I think, around 1.5 millimeter. That's very impressive. A very, very long spirostom. So I think we've covered everything that I wanted to show you today. I see there are no questions, so we're gonna sum it up. I wish you a good night and hope we'll have another episode soon. Good evening.